So chapter 14, we're going to be talking about transcription, which is the synthesis of RNA. Here are your learning objectives again. There's only 7, but each of them are pretty in-depth, so make sure you can answer them fully. So here we have a few terms that relate to RNA synthesis. Make sure you know them since it is your first learning objective. So template strand is the strand that we're going to be using as a template to make new RNA. The coding strand is just complementary to the template strand, and another name for the coding strand is called the non-template strand. So here's a picture relating the coding strand and template strand. So remember, the coding strand of DNA is just complementary to the template strand. We're going to be using the template strand to make our new RNA. So if you look at the mRNA, it's going to be complementary to that template strand. Also notice, since we're using RNA, we're not going to use thymine as a base pair. We're going to use uracil. Another definition is the promoter sequence. It just identifies the start point so that RNA polymerase can bind there and start RNA synthesis. So just another thing to pay attention to is in the promoter region is a sequence called the Tata box, and that's where starting point of transcription begins. So a few more definitions to know. Primary transcript, hRNA, is the first RNA form produced. Upstream of transcription means the left of the starting site, so towards that 5' end, and downstream of the transcription is to the right of the start site, so it'd be going to the 3' end. So here's a picture of the prokaryotic and eukaryotic promoters. At the top we have a prokaryotic promoter. So notice the Tata box, that's going to be in your promoter region, and that's where RNA polymerase will bind. There's also activators, which stimulate transcription, and repressors that inhibit transcription. At the bottom, we have a picture of eukaryotic promoters. So the Tata box is there. That's where the RNA polymerase will bind. And then there's this yellow box called promoter proximal elements, and that's just a binding site for different transcription factors that can accelerate the rate at which RNA polymerase binds to the promoter. So here's a picture of RNA synthesis. If you look at the structure of UTP, you'll notice that the alpha phosphate is what's going to connect the ribosyl groups together. So here's the slide we saw earlier. It's just relating the coding strand, template strand, mRNA, and protein. So coding strand is 5' to 3'. Your template strand is your 3' to 5'. And your template strand is going to produce the mRNA. So the mRNA is in sequences of 3. So those are called codons, and each codon is going to specify a certain amino acid. So that's when they make the proteins. So these are the type of questions you should expect to see on the test. Deduce the DNA species from which the following mRNA was derived. So make sure you read these questions carefully. The question is asking you to find the DNA strand that this following mRNA strand was derived from. So in the blue, that's your mRNA strand. We know that the RNA strand is made from the DNA template strand. So our answer is going to be the DNA template strand. That's what we're trying to find. So we know that the template strand is complementary to the mRNA strand. So the easiest way to figure out problems like this is to just write down that mRNA strand, so that's the one in blue. Right above it, write from 3' to 5' since it's anti-parallel, and then write the complementary base. So you should get C, C, G, C, T, A, C, T, A. So that's the answer from 3' to 5', but since he has the answer from 5' to 3', you just have to flip it over. So the answer would be B, which is A, T, C, A, T, C, G, C, C. So just practice these sort of problems a little bit, and you should be fine on the test. So this is another learning objective, number 3, identifying the function of the following transcription signals. So you should know all of these really well. You have your proximal elements, which is going to be upstream. So remember, upstream is left of the start site. And those contain your regulatory elements, so things that could accelerate transcription. Then you have your distal elements, and they're very similar to proximal elements, except they're farther upstream, so they have a weaker influence than the proximal elements. Then you have cis-acting elements, which are responsible for gene expression. You have your trans-acting factors, which can bind to those cis-acting elements to control your gene expression. Then you have enhancer elements, which are going to enhance transcription, and silencer elements, also called repressors, which are going to inhibit transcription. So here are the transcriptional signals, sort of gives you a visual of where they're located on the strand. So here we have our transcription signals once again. We have our activators, also known as enhancers. Those are located at a distant site on the DNA. And so when DNA folds, those activators are going to send a signal to co-activators, and co-activators are going to transmit that signal and send them to basal factors. So basal factors, in response, are going to position the RNA polymerase at the start site. So all of these activators, co-activators, and basal factors are going to enhance transcription. Repressors and silencers are going to slow down transcription. So prokaryotic transcription is pretty similar to eukaryotic transcription. It uses a template strand to make RNA, and then RNA, it's going to produce proteins. So prokaryotic genes are organized as transcription units. The RNA polymerase is going to bind also to the promoter region, so that's where transcription is initiated. Once that occurs, transcription bubble is going to be created. RNA polymerase does not proofread. Synthesis is always in 5' to 3' direction, and termination is signaled by a hairpin loop, which is going to release the RNA from the transcription bubble. So these are the products of eukaryotic RNA polymerases. You should know these very well. So RNA polymerase 1 is going to be rRNA. RNA polymerase 2 is mRNA and microRNA. And RNA polymerase 3 is tRNA and other small RNAs. So make sure you know those. So in our promoter region is where we're going to find the TATA box, and that's where your RNA polymerase is going to bind to initiate transcription. So TBP stands for TATA binding protein. So that protein is going to bind to the TATA box. As soon as that happens, transcription factor 2A and transcription factor 2B are going to bind to the TATA binding protein. Then your RNA polymerase will come along, and your other basal transcription factors, TF2B, F, and H, are going to bind to that RNA polymerase. Cofactors can also bind to it to accelerate this process. So this is just a structure of bacterial transcription initiation complex. Just read through to see what it's formed by. So here's just an overview of transcription. Remember, we're using the DNA template strand to make your RNA transcript, and the direction of transcription is 5' to 3'. So after you make that hnRNA, the other 3 steps are 5' capping, a poly A tail addition, and then removal of introns by spliceosomes. This is the same picture we saw earlier. It makes that hnRNA strand. There adds a 5' cap, a poly A tail, and then we remove the introns using spliceosomes. So transcription is going to produce your hnRNA from your DNA template. And after hnRNA is produced, there's an addition of a 5' cap and a poly A tail. So we use splicing to remove introns, which is going to give us our mature mRNA. And once we have that, it can leave through the nuclear pores and into the cytoplasm, where it's going to make proteins. Here it just shows you the cap structure in eukaryotic mRNA. So this is a picture of the poly A tail. Your RNA polymerase is going to pass a polyadenylation signal, that's AAU. Once it passes that, it'll reach a cleavage site where it's going to cleave off the remaining nucleotides, and a poly A polymerase is going to come in and it's going to add 250 adenine nucleotides to the end of that 3' end. So these are the splice junctions in hnRNA. So before we make mature mRNA, they're going to contain regions known as exons and introns. So we have to remove the introns because they don't contribute to the amino acid sequence. And you can tell that it's an intron because almost all introns begin with a 5' GU and they end with a 3' AG. So exons are going to be spliced together so that the appropriate protein is produced. So this is the splicing process. We have the exons in blue boxes, and we have the introns represented by the pink strand. So we want to keep the exons and remove the introns before we go through translation. So remember, the start of an intron is going to start with GU, and it's going to end with AG. So that's where the SNRPs know where to bind. So U1 is going to bind to GU. U2 is going to bind to an adenine nucleotide within the intron, and U1 is going to loop around and bind to that adenine nucleotide. So at this point, the other SNRPs, U4, U5, and U6 are going to bind together, and they form this complex called the spliceosome. So the spliceosome is going to remove your intron, and notice it's in a lariat form, so that's just going to be degraded. And then your exon 1 and 2 are going to be joined together. So this again is just another picture showing you the same thing that we just looked at. You have your GU, which is the start of your intron, your AG, which is the end of the intron, and then that adenine nucleotide where U2 binds to. So objective 5 is knowing the classes of pre-mRNA splicing defects that cause disease as well as splicing-related diseases. So here's a picture of classical and auxiliary splicing signals. In A, we have the classical splice sites. In B, we have splicing elements and binding factors. And in C, we have cis-acting splicing mutations. On the next few slides, we'll talk about the diseases. So here we have classes of pre-mRNA splicing defects that can cause disease. So we have cis-acting mutations that disrupt the use of constitutive splice sites, and we have the cis-acting mutations that disrupt the use of alternative splice sites. So just be sure to read through them to differentiate between the two. So here are your other two classes of pre-mRNA splicing defects. So we have 4 total. This is transacting mutations that disrupt the basal splicing machinery, and then we have transacting mutations that disrupt the splicing regulation. So again, just really know the difference between all 4 of them. So here's a very important slide. He lists some human diseases that is caused by disruption of the pre-mRNA splicing. So be sure to read through these different diseases and know what can cause them. So bacterial genes are organized into operons. So operons are things that can have a single promoter region, so a single promoter, but it can encode for multiple proteins. So one promoter in a bacterial operon can encode for protein A, B, and C. So here's just some reasons why humans don't have operons and why bacteria do. So just be sure to read through these. But this is just a picture of the hypothetical human chromosome. So this slide talks about eukaryotic rRNA and ribosome synthesis. So eukaryotes, most of the rRNA is going to be transcribed from DNA, and this matures in the nucleolus. Other key things to point out is that the 40S and 60S ribosomal subunits are going to assemble separately in the nucleus, and you'll see a picture of this later. And then the ADS is going to be assembled in the cytoplasm, and it's assembled by the 40S and 60S ribosomal subunits. So this is a picture of rRNA and ribosome synthesis. You can see that the ADS ribosome is being made in the cytoplasm, and earlier we said the 40S and 60S are going to be made separately. So you should know the precursors of the 40S and the 60S. So for example, 60S is coming from the 28S, the 5S, the 5.8S, and then those are going to come from the 32S and the 5S, and then that's going to come from the 41S and 5S. So just be able to draw those out for me if I ask. So here's just a different way of showing the animation. So the 45S RNA becomes the mature 18S, 28S, and 5.8S. So really just know the precursors that make everything up. This is just a picture that shows you by the arrows where they're cleaved at to produce the 16S, 23S, and 5S rRNA. So to make tRNA, we need a precursor of 100 nucleotide strands. Remember, tRNA is that clover leaf shaped structure. RNase P cleaves that 5' end, and if introns are present, they're going to be removed by endonucleases and ligated by RNA ligase. Some of the bases are going to be modified during the cleavage reactions, and there's going to be a CCA addition at the 3' end before the tRNA can leave the nucleus. This is just a picture showing you that the green regions are the promoter regions for tRNA transcription. Here's an overview of tRNA synthesis. We have to make it from a DNA template strand. It forms that clover leaf structure. If there's an intron, we remove the intron. We modify the bases and add that CCA tail, and then it can leave through nuclear pores into the cytoplasm. So here is another learning objective. Just be able to describe these below. We'll talk about them in the next few slides. So some mushrooms have alpha-amanitin in it, and alpha-amanitin is an inhibitor of RNA polymerase II. So there's no cure for this, and between 40 and 90% of people who ingest alpha-amanitin will die within a few days. This is just the structure of the RNA polymerase II that gets inhibited by alpha-amanitin, which comes from some mushrooms. So the key thing to know from this slide is rifampin is a drug that inhibits bacterial RNA polymerase. So if we have a bacteria infection, rifampin can selectively kill the bacteria since it inhibits that RNA polymerase. One thing to know is that rifampin does not affect eukaryotic cells. So it is selectively killing the bacteria. So here we have two drugs, actinomycin D and acridine. Both of these are going to inhibit prokaryotic and eukaryotic transcription. So when your body is infected by a virus, certain things are going to happen. TAT, REV, and NEF are going to be produced. So TAT stimulates transcription of viral DNA. REV causes those RNA transcripts to leave the nucleus unspliced. NEF indirectly aids in assembly of the viral particles, and then GP41 and GP120 are envelope proteins that enter the cell membrane, and POL is a reverse transcriptase produced from viral RNA. So here's the life cycle of HIV again that you saw in the last chapter. One of your learning objectives is to compare their mechanisms of actions with the inhibitors, rifampicin and alpha-amanitin. So just be able to tell me how the drugs work and how they're different from each other. So this slide talks about how HIV can enter the cell. So in order for HIV to enter the cell, it has to bind to a CD4 receptor and chemokine co-receptor, CCR5. So in individuals who carry a mutation in the CCR532, it's going to prevent HIV from entering the cell. So CCR532 is a 32 base pair deletion, so it's a mutated gene. So 10% of Europeans are going to be resistant to HIV because they have that CCR5 mutation. Here's your disorder table. Be able to identify the different diseases and their conditions. And just last minute tips is try try to watch YouTube videos if you have trouble understanding any of the processes, get a visual understanding of it, and if you can, try to get at least one study buddy so that you can try and explain these different processes of DNA replication, transcription, and such. So good luck, guys.